is Angus Sinclair. Angus is known for a long time in the field. He worked in Cambridge, San Diego, and then Southwestern Medical Texas in academia, switched to large and small biotech and pharma companies, including Amgen, GM Biosciences, and Northern Biologics. He's now the CSO of LabGenius Therapeutics, really exciting companies, and he is going to tell about engineering of T-cell engagers for complete on-off killing selectivity by, again, machine learning and high-throughput experimentation. So I'm really eager to learn how much experimentation and how much machine learning has been involved. Thank you.
Great. Well, first of all, I'd really like to thank the organizers of PEGS for inviting me to present the data from LabGenius today at PEGS Europe. So it is fantastic to be back at PEGS Europe, but also to be in Lisbon. So I'm going to tell you about the work that we're doing at LabGenius to engineer T-cell engagers for solid tumor indications that we believe have complete on-off killing selectivity to really mitigate some of the on-target off-tumor toxicity that's seen with a number of T-cell engagers. And this is through using our machine learning and high-throughput experimentation systems called the AVA platform. So just as an agenda for my presentation today, I'll give you a bit of an introduction about LabGenius and the AVA platform, and then go into a case study where we utilized our technology to develop highly specific HER2 by CD3 T-cell engagers with high degrees of selectivity. And this is just a case study. However, we have a lead asset where we haven't disclosed the target as yet, again, targeting solid tumors, where we've also been able to engineer this high degree of selectivity to really mitigate the on-target off-tumor toxicity. And I'll also provide some pilot in vivo screening data as we advance this molecule into IND-enabling studies. So as you are well aware, immunotherapeutics in some cancer indications have had significant benefits, such as checkpoint inhibitors in certain lung indications, et cetera. However, there really is still an unmet need for therapeutics to have durable responses in patients. T-cell engagers are a very exciting class of molecules to try and obtain these durable responses. And this has obviously been seen in the heme malignancy space, where T-cell engagers have had tremendous success. However, in the solid tumor setting, this really has been challenged by a number of factors, which include on-target but off-tumor toxicity. And because of the solid tumor space, there really are very few selectively upregulated tumor-specific antigens, so often investigators need to evaluate targets that are upregulated on tumors but still are expressed at low levels on normal healthy cells of tumor origin. And this just illustrates some of the challenges that are faced in designing really the highly specific T-cell engagers in the solid tumor space. And this cytotoxicity plot just shows you maybe a conventional T-cell engager that is able to very potently kill cancer cells on the left-hand side, but still has residual activity on normal healthy tissues. So this therapeutic window is really narrow, and often though these therapeutics may have been successful in the clinic, they're really limited by the doses that are able to be obtained because of that toxicity. There have been various conventional approaches tried to try and improve this therapeutic window, such as downregulating or reducing the affinity of the binders to try and sort of mitigate between targeting the higher levels of expressing tumor cells in comparison with the lower level activity against healthy cells. However, some of these still continue to have challenges, again, not being able to meet those high doses needed for solid tumor efficacy. At LabGenius, we've developed a platform called AVA that we believe is the only platform to really be able to deliver complete on-off killing selectivity, where we're able to really dial in the activity of the T-cell engager to very effectively and potently kill cancer cells, however, really have minimal to no activity on normal, healthy tissues. And these can be down, and I'll show some examples, even down to 2-fold levels of expression of target. So how are we going about to really dial in this selectivity? We are utilizing the avidity of the molecules to be able to fine-tune being able to target the higher levels of expression tumor cells, as well as not be able to target the lower expressing healthy tissues. And this example just highlights on the left-hand side tumor cells that have higher levels of target, and on the right-hand side, healthy cells that have lower target. And we really, if you have a high affinity or high valency of your binders, are really able to kill both cells. So really is not being able to dial in this type of activity. However, if you have lower affinity and valency of your T-cell engagers, really you're taking away the potency of those molecules, so really disabling those molecules from being able to be highly effective. So we don't believe that this is the right approach to modulate the activity of our T-cell engagers. So how we are going about doing this is by using the affinity and the valency to be able to fine-tune the molecules to have that therapeutic window where we're able to see high degrees of killing on cancer cells, but really remain the healthy cells untouched. So I'll just provide an introduction to our machine learning and automation platform called AVA, as well as a bit of an introduction to LabGenius. So we are currently based right in the center of London, and we have a facility about 4,000 square foot where we have high throughput automation, where we utilize the high throughput automation really from the start of cloning, being able to produce our proteins in mammalian expression systems, as well as purifying those molecules at a relatively low scale, that we can then ultimately do our experimental analyses, and we include developability of these molecules within the high throughput automation process, as well as evaluate cell-based cytotoxicity assays. So we look at multiple different parameters to try and really optimize these T-cell engagers on multiple levels as we are doing the experimentation. As you're well aware, antibody discovery is very hard because often, if you're modulating one property of the antibody, such as improving affinity or expression, that you can ultimately introduce additional variabilities that ultimately don't result in maybe a stable or a highly manufacturable molecule. So we systematically and in parallel optimize the efficacy of our molecules, looking at how potent those molecules are and how specific is the drug, as well as balancing the safety, evaluating the toxicity and immunogenicity of our potential therapeutics. As mentioned, in parallel, we evaluate the manufacturability using early developability processes to ultimately understand how well this molecule may be taken forward in GMP processes. as well as evaluate the stability of these molecules. So we're really navigating multiple parameters as we improve the generation of our molecules. And we take a lot of different factors into consideration when we're beginning to design our molecules. We look a lot at linker length and linker rigidity. It's interesting, whilst you may have a very effective and highly selective molecule, just by changing one linker, you can really reduce the potency or reduce that selectivity. We evaluate the affinity of the binders to the TAA, as well as look at multiple epitopes that may be optimal in the design of the molecules, as well as evaluate multiple valencies and topologies of the molecule to really understand, when we introduce the CD3 binding domain, how does that best influence the T-cell mediated cytotoxicity, as well as dial in that highly selective nature of the T-cell engager. So we first of all start out with the building blocks of these molecules. So in the red, we select a large number of humanized, diverse binders for different epitopes with a very broad range of affinity. We don't, at this point, really sort of narrow our scope. We really want to go in with a very broad range to help the machine learning and the automation to tell us what are the most valuable properties for those molecules. We generally use VHHs, but we also utilize single-chain FVs in the design of these molecules. As mentioned, we also utilize linkers of various lengths and flexibilities because we do identify these introduce different properties into the molecule. And we fuse these to a silenced FC for half-life extension, as well as removing any residual effector function. So using these building blocks, we introduce this into the AVER platform, which designs a vast number of molecular designs. We are up to about 2 million designs that we can implement in the designing of a potential therapeutic. And these are evaluated both functionally and developability. But obviously, we're not evaluating 2 million molecules. We do random sampling, which we call explore. And then once we see molecular properties being introduced that are favorable for our molecules, we then exploit some of those properties to further refine to get the optimized T-cell engager, as illustrated on the right. So our format is really well suited to multivalent and multi-specific design requirements. So some of the key features that we're really optimizing in these molecules are obviously selective tumor targeting. We evaluate bivalent as well as trivalent TAA binding to really dial in that selectivity. We utilize unique geometries, as I mentioned, sort of positioning the CD3 binder in various different locations to understand the properties of those molecules and the various linker combinations that design or provide the properties that we desire. We also have a monovalent binding to CD3 to reduce CRS. However, that is included in our molecular evaluation as we move forward in the analysis and lead optimization of these molecules. We generate these for also subcutaneous dosing, so we want molecules that have flexibility in their dosing regimens, as well as obviously favorable half-lives that could be used for every 2 to 4 week dosing. So we utilize an iterative process called active learning to really evaluate the antibody design space. And we really have this separated out into the dry lab and the wet lab aspects. So in the dry lab, as I mentioned, we can design in silico about 2 million antibody designs. Using not only from de novo designs, but also maybe data that we have generated through our machine learning cycles. We then sample around 2,300 molecules from that initial design space and move those into the highly automated system to clone and produce these molecules and include developability to try and filter out molecules that don't have a potential path forward in this process. We then move on to the functional characterization of these molecules. So we try and use as close as possible to physiologically relevant cell-based assays that represent either the tumor levels of expression using cell lines equivalent to the tumors, and we often do internal assessments of those levels, as well as the healthy cell levels. And we usually use 2 to 3 cell lines in our high-throughput screening process, and usually use 2 to 3 PBMC donors. So we're using primary human T cells to measure the activity of our molecules. Once we get the data from a particular cycle, we feed that back into the machine learning process. So we're able to look at the properties introduced or the data that come out from these multi-parametric values, and then look at the properties and then feed that back into the design so we can begin to further explore or exploit the molecular profiles that ultimately introduce the therapeutic properties that we would like. And this takes about less than 6 weeks to do a complete cycle from the design to being able to interpret the data and then feed that back into additional designs. And as I mentioned, in the wet lab, we currently have a high throughput system of about 2,300 molecules per cycle in the experimental analysis. So first of all, I'll go through a case study where we evaluated the potential of generating a highly selective HER2 by CD3 T-cell engager. And in this particular case study, we were able to get molecules that had about 400 times better selectivity than a clinical candidate, rototumumab. And I'll show you some of the data with regards to that molecule. So this was a while ago, so we had a much smaller design space, so we evaluated about 44,000 different designs, utilizing 48 VHH binders to HER3, evaluating valencies in the 1 to 3 order. We only had a single CD3 binder in this particular instance, but we had 10 different linkers of various lengths and flexibilities that we introduced into the molecular designs and ultimately explored in our cell-based assays. We also evaluated multiple different topologies of those molecules as well, looking at, as I mentioned, the placement of the CD3 binding domain, not only C-terminal, N-terminal, but internal to bivalent and then also trivalent molecules. So just before going through the data, I'll explain the axes on these various graphs. On the left-hand side is the T-cell activation data set. So at this early point, where we're using reporter T-cell activation-based assays in this case study. On the y-axis is T activation score, so higher is sort of more potent molecules. And on the x-axis is a T-cell activation selectivity score. So in this study, we used 2 cell lines, one that was representative of tumors. They're expressed around 300 to 400,000 copies per cell of HER2. And then for the healthy cell equivalent, we had a cell line that expressed about 25,000 copies that would be equivalent to a cardiomyocyte expression levels. On the right-hand side, and you'll see as the plots build, we took the top 25 molecules per cycle and then built in sort of a compound metrics, evaluating both the activity of the molecule, but also the selectivity of the molecule to really understand which would be the therapeutic properties that we may want to take forward. In the blue, in our first cycle, as you can see, we really didn't have a lot of selectivity, but a lot of different potencies. And as we went through 5 different cycles of this machine learning and wet lab assessment, we were able to build and expand upon the selectivity of our molecules. So ultimately resulted in a molecule that had significantly better selectivity than the ranibizumab benchmark control that we were comparing it to. And this took about 5 cycles to generate these data. So as these are reporter cell-based data, we really wanted to understand from a more physiologically cell-based assay, how would these perform? And these are just some plots indicating the T-cell dependent cytotoxicity, again on the high HER2 expressing cell line in the darker plots on the left-hand side, and the healthy equivalent cell on the lighter, either blue or the pink plots. The top left-hand plot is looking at the benchmark control. And as you can see, there's about a 30-fold window between killing tumor cells as well as killing what we would consider healthy cell levels. We were able to identify multiple molecules that had very high degrees of selectivity in the order of 10,000-fold in comparison with healthy cell levels. So again, being able to identify these molecules with these high degree of selectivity without really introducing any human bias into the interpretation or the design of these molecules. So in this particular case study, we were able to start out with about 44,000 designs, and then really through our machine learning and sort of sampling that explore and exploit approach, really only test about 980 molecules in the wet lab to be able to identify the properties to provide those highly selective T-cell engagers that had a potential for best-in-class selectivity profiles without losing potency on the cancer cells. So we currently have a lead asset that is about to enter into IND-enabling studies. We haven't disclosed the target for this particular T-cell engager. But again, we have been able to identify highly selective molecules that really are able to differentiate between tumor and healthy tissues, even to levels and pushing the envelope about 2 levels in different levels of expression. So in this case, we started out with a much larger design space. So about 270,000 different molecules were designed in silico utilizing 25 binders to our target, and these were VHHs, looking at valencies of 1 to 3. In this particular instance, we used 3 T-cell CD3 binders, but again, just retained this to 1, and then 20 unique different linkers, again, in various lengths and flexibilities to provide this, this complex molecular design. And here we utilized 3 cell lines, 2 cell lines that had what would be considered tumor equivalent levels of expression, about 25,000 copies per cell, and then a cell line that had what would be equivalent to the highest levels of expression in a normal healthy cell, about 5,000 receptors per cell. And we utilize these cell lines in our high throughput automation cell-based T-cell dependent cytotoxicity assays. These plots just illustrate 3 of our potential lead candidates and the molecular profiles or the cytotoxicity profiles of these candidates. In the black is the cytotoxic activity on the tumor cell with about 25,000 copies. And in the pink is the cytotoxic activity on the healthy cell equivalent, about 5,000 copies. And as you can see, we have very, very high degrees of selectivity where we're really not seeing any cytotoxicity of that normal healthy cell equivalent. As mentioned, we really pushed the envelope to see if we could find out how far could we get, and we were able to get sort of selectivity on cell lines that expressed about 2 times the levels of the healthy cells. Though the potencies of some of these molecules were similar, the Emax was different. So we might be hitting a threshold for our molecular designs in this particular instance. We have just run some in vivo screening assays to really select the candidates to move forward into IND-enabling studies. And these were very small studies, small numbers of mice, but I just wanted to whet your appetite with a little bit of in vivo data on the left-hand side, looking at tumor growth inhibition. We were able to get up to 70% tumor growth inhibition in these relatively short-term models, because these are limited by the onset of graft versus host disease, because these are human PBMCs in immunocompromised mice. And on the right-hand side, we were able to see significant tumor growth regression in some of these mice. We're now conducting follow-up studies to expand upon these data set and really understand the dose responses that we see with this molecule to further enhance and moving forward these into IND-enabling studies. So we currently have a pipeline of internal assets. Our lead candidate is just about to enter into IND-enabling studies. We're also expanding the platform to look at tri-specific T-cell engagers as well, and really leverage our AVA platform to really identify molecules with that desired selectivity and build in, some of the resistance mechanisms that tumors may downregulate, certain targets and be able to, still have activity of our molecule. But we're also exploring ADCs as well, as there are a significant number of ADCs that have been approved that still have on-target but off-tumor toxicity. So we are exploring this space as well, as we're also very interested in establishing collaborations with potential partners that may be interested to explore other modalities, because these are really well suited for our platform to expand and explore really some of the challenges that face sort of solid tumor targeting therapeutics. So with that, I'd like to acknowledge sort of all of the teams, past and present, that have contributed to getting us to this point. It's a real tour de force, and very happy to take any questions that you may have around our therapeutic platform. Thank you.